plants and circulation in animals. So then before class we see the transportation in plants, how the transport the minerals and waters are transport to the each and every part of the plant. Now we are going to see the circulation. So circulation, so here in this chapter we are seeing blood. In an animal, the circulation is taking place through the blood. So the blood is a main circulatory member in the human body. It is red colored, connected to the tissue, connective tissues are there. So in this blood, what are the components in a blood? There are two main components are there. So one is a fluid, that is a plasma. Another one is called formed elements, that is a blood cells. So it is suspended in a plasma. So when we see in a plasma, it is slightly alkaline nature. It are cellular non-cellular substance which contain about 55% of a blood. So this organic substance that lies protein, glucose, urea, enzymes, hormones and vitamins and minerals are present in the plasma only. So next is the element, that is elements of a blood is corpuscles. There are three types of corpuscles are the RBC, otherwise called the red corpuscles or erythrocytes, WBC or white blood corpuscle, otherwise called as a leukocyte, blood platelets or otherwise called the thrombocytes. So first we go to RBC, that is otherwise called the erythrocyte. So most abundant cell in a human body. So this RBC is formed in a bone marrow. It is red in color. It is help for respiration. So how it help? It having a pigment that is hemoglobin. So it help for respiration, and also this hemoglobin is present in this cell. So it is a red in color. So matured mammalian RBC have a cell organelles and also nucleus. They are biconcave and a disc shape. In this picture you see how the cell is present. It's a biconcave and a disc in nature. So in this RBC lifespan is 120 days. It is formed in a bone marrow and the lifespan of RBC is 120 days. So what is the function of this RBC? Transport oxygen from a lung to tissue. Next we go to see WBC or a leukocyte. So leukocyte, it is a colorless thing. So RBC red in color, in WBC it is a colorless, but you are asking why in a picture it's giving a violet color or a pink color. So when you see the cell under the microscope, we want to give any dye, it is easily identify the cytoplasm, nucleus, all the thing. So what the cytoplasm, nucleus are absorbing, that color will be exposed in a picture. So the white blood corpuscles are colorless in nature. Why it's colorless? It's not having a hemoglobin and other different between RBC and WBC. So RBCs are non-nucleated, but WBCs are nucleated. So RBC we are seeing nucleus not having a cell organelles and also nucleus, but WBC you see a cell organelle nucleus also we see. So in this WBC also formed in a bone marrow. Bone marrow, spleen, thymus, lymph nodes, all these area is found at this WBC. It's capable of move. It is move like a amoeba. So amoeboidal movement we are seen in a WBC. So WBCs are classified into two categories. One is a granulocyte, another one is a agranulocyte. In a granulocyte, the granules in the cytoplasm, they are irregular and lobed in nature. Which is irregular and lobed in nature means nucleus. So three types are there in a granulocyte. One is a neutrophil, another one is a eosinophil, another one is a basophil. In this picture you are easily seen neutrophil. So neutrophil, it is a lobed nucleus we are seeing here. 3 lobes are there. In basophil 2 lobes are there. And lymphocyte we are seeing here. Lymphocyte there are no this. Then in basophil you are seeing this violet, dark violet in nature, lobe-like nature, it is called as a basophil. 3-lobed means or many-lobed means neutrophil, 2-lobed means eosinophil, slightly lobed means it's basophil. So in this neutrophil, large size, this totally all the cells, this neutrophils are large in shape. It is 60 to 65% of total leukocyte. It is a leukocyte cells only. So you are seeing 2 or 7 lobe in this leukocytes, neutrophils. Their number are increasing during infection and inflammation. So 60 to 65 normally we seen in a overall leukocyte, that is normally we see 60 to 65% of overall leukocyte we see neutrophil, but infection we taken electron microscope, see the sample of a blood, many neutrophils are there. Next we go to eosinophil. Eosinophil is a blob. It is also bilobed nucleus and consists of 2 to 3% of total leukocytes. Number of infection increase during allergic reaction. So in our body having a allergy or parasitic infection, automatically we are seeing a percentage of eosinophils are more in a blood. Next we go to basophil. Basophil have also lobed nucleus. They are 0.5 to 1% in a total leukocyte. They are releasing chemical during the process of inflammation. When your body is attacked by any damage, that region will inflamed. We are taken a blood sample in that area, particular area. The basophils are more in nature. These basophils are producing a chemical during a inflammation time. So next we go to A granulocyte. The granules are not found in the cytoplasm. The A granulocytes are 2 types. It's a lymphocyte and monocyte. You are seeing this. There are no granules. Dots are here. So only nucleus are seen in a cytoplasmic region. So it is a lymphocyte and monocyte. So lymphocyte, total our body 20 to 25% of total leukocyte. We are seeing a lymphocyte. They produce antibodies during bacterial and viral infection. Monocytes, they are also largest leukocytes of the amoeboid in shape. They are cells formed 5 to 6% in total leukocytes. They are phagocytic and can engulf the bacteria. Phagocytic nature. Then when the bacteria is entering in our blood, automatically the monocytes are engulfed this bacterial cell and degenerate the bacterial cell. So this is the main function of. So our WBC cells, white blood corpuscle cells, are used for immunity function. Next we go to blood platelets or thrombocytes. This is a small colorless structure in a WBC, in a blood cells. They do not have a nucleus also. So totally 250,000 to 4 lakh cells we are seen in a blood, these blood platelets. So in this lifespan of blood platelets are 8 to 10 days. They play an important role for clotting of a blood. The platelets are form a clot on the site of injured and prevent the blood loss. So these blood platelets, thrombocytes, doing a function that is called as a prevent a blood loss, and also during injury it can form a clot. So where you are injured or broken or skin will be peeled, we are seeing a small blood is came, after that will be clotted. ஒரு கட்டியான ஒரு things we நம்ம பார்க்குறோம். So in that clotting function is done by of the blood platelets. So only we are seeing the lifespan of cells, the blood cells, only RBC and also the platelets. RBC 120 days, platelets 8 to 10 days. But WBC never seen a lifespan, because when it's injured or damaged or any bacterial or viral infection, automatically it will start to fight and it will be disappeared. So what is the function of the blood cell? So transport of respiratory gases, oxygen and carbon dioxide with the help of RBC, then transport of digested food materials from different parts of the body cell, and transport of hormones, and also it is transport nitrogenous waste like ammonia, urea and uric acid, these things are transported. So next it's help for WBC cells are used for defense against disease like all the disease. Then it is act as a buffer for regulating pH of body temperature, and it's maintain our water balance in our body. So these are the main function of our blood. So next we see the circulatory. It is when you are thinking a circulatory in a circulation in our body, one organ is very important. So one system is very important, that is our human heart. Structure of human heart. So human heart is a muscular pumping organ. So it is blood, this pump out the blood into our blood vessels. So human heart is situated in between the lung. It's slightly tilt towards the left, and it is present above the diaphragm in a thoracic cavity. It is the human heart is made up of special type of a muscle. It is called a cardiac muscle. So it encloses a double-walled membrane. It is called as a pericardium. So in this it contains lubricated in between the two membrane, a pericardial membrane, the space is there. It is filled with a fluid. It is called as a pericardial fluid. So in this pericardial fluid, it gives reduce the friction during heartbeat and also product the mechanical injury to our heart. So this human heart having 4 chamber, 2 chamber are thin walled chamber. It is called as a auricle or atria, and it's a 2 lower chamber. It is a thick walled chamber. It is called as a ventricle. These chambers are separated by a septum. So each septum or auricular ventricle prevent mixing of oxygenated and deoxygenated blood. So these 2 auricular separated from other, it is called intra-auricular septum. So it is separating left atrium smaller than the right atrium. So right atrium is receiving deoxygenated blood, different part of the body, and its main veins are superior vena cava, inferior vena cava, and coronary sinus, and pulmonary vein brings to oxygenated blood in the left atrium. So it's right atrium, left auricles pump the blood, and right and left respectively. So the ventricles form a lower part of the heart. It is ventricle is separated by a septum. It is called intraventricular septum. So this right ventricle have a thick wall because the ventricles have a pump of blood in the force of the heart. From the right ventricle, the arise to the pulmonary trunk. So the bifurcated the right and left pulmonary artery receive the pulmonary artery supply deoxygenated blood of the lung and respective sides. So pulmonary trunk will be, you are seeing this picture how that membranes are there and separated by septums. So right atrium, left atrium, right ventricle and right ventricle. So the pulmonary trunk taken and come and attached to the our right ventricle. So pulmonary vein, it receiving oxygenated blood. These oxygenated blood are enter into our left atrium. So left atrium having oxygenated blood, and right atrium and ventricle having a deoxygenated blood. So you are seeing superior vena cava, aorta, pulmonary trunk, pulmonary valves are there. So cardiac muscles are also here we are seeing. The inferior vena cava. So inferior vena cava and superior vena cava. These two will be taken deoxygenated blood through all the part of the body and enter into our superior and inferior vena cava entering into our right atrium region. So next it's the respective part will be entering the blood vessels. So next this part what we are seeing here, it is called as a valves. So left ventricle gives the rising aorta. It is a left ventricle gives rising aorta. It is called as a this aorta supplying oxygenated blood throughout the body. So we are seeing here the superior and inferior vena cava taken deoxygenated blood and pour into the right atrium. It entering the left atrium and passes to the pulmonary trunk. Then oxygen will be in a lung, it's entering. Automatically lung will be remove the deoxygenated blood and converted to oxygenated blood. It receiving and coming to the pulmonary vein, and it's entering in the left atrium and pass to the left ventricle. Left ventricle to through the aorta, it passing throughout the body. This method the blood will be circulated in a heart. Next, in these blood vessels we are seeing a valves. So the valve or a muscular flap that regulate the flow of a blood in a single direction to prevent the backward direction. So here three valves are there: right atrioventricular valve, left atrioventricular valve and semilunar valve. Right atrioventricular valve, where we are seeing here right atrium and right ventricle, in between the septum is there. This will be covered by right atrioventricular valve, or otherwise called as a tricuspid valve. So in this tricuspid valve, region of this ventricular muscle, this contraction is spread to the ventricular muscle. It's called arterioventricular bundle, otherwise called as a Purkinje fibers. These three region is given contraction and relaxation of the heartbeat. So started from SA node. The SA node is otherwise called as a pacemaker. So what is a pulse? The heartbeat of the blood forced into 8 arteries. It expression of this artery every time of the blood force is called as a pulse. When the blood will be passed to the blood vessel of artery, so it giving a pulse. So we felt the placing of fingertip on the artery near the wrist or any main small arterial region, you feel that pulses region. So normal pulse rate range is 70 to 90 minutes. Heartbeat is 72 to 75 minutes, but pulse rate is 70 to 90 minutes. These two are different. Heartbeat is happen for contraction relaxation of a heart, but in the pulse will be the blood force it creating a pulse in our body. So now we finished the lesson, that is transportation in a plant and circulation in a animal. So how the bloods and ions are move in a plant, and how the blood is present, blood components are there. So what are the blood components are present in our blood. So next we say the two, then plasma and blood cells are there. So blood cells, three types of cells are there: RBC, WBC and platelet. What are the functions of RBC? What is the function of WBC? And what is the function of platelets? Are the total function of blood also we see. So what is the structure of human heart? Then what is the how the heartbeat is present, how the pulses are happen in our body. All the things we are seeing. Next we are going to see a blood group. So blood group is the grouping of a blood is developed by Carl Landsteiner. So he given a three type of a blood group, A, B and O, and AB blood group is recognized by staining. So this agglutation or antigen antibodies, these are present in RBC, WBC. Depend upon this they are grouped into 4. So and this RBC surface membrane, RBC membranous surface, the antibodies are presented. They are called as antigen A, antigen B, antigen C, A group, B group. So how they are present? So in a A group, individual antigen A is present in a RBC surface. So the blood group is called A blood group. Antibody is present. Presence of antigen in our RBC surface, based on that we are classified 4 groups: A group, B group, AB and O. So A group means antigen A is present on the surface of RBC, antibody B is present on the plasma. So that group is called A. So antigen B is present in the surface of RBC, antibody A is present in a plasma, that group is called as a B group. So antigen A and B is present on the surface, no antibodies are present in the antibodies are absent in the plasma, it's called AB blood group. Antigen A or B absent in the surface, antigen A is also absent, B is also absent in RBC, but plasma contain both antibodies, they are called O group. So blood donate, how we want to give a blood? So we mismatchly given A group and B group to the recipient, so automatically agglutation, clumping is happen. So it leads to death. So we want to match the compatibility of antibody antigen in a blood sample. After that only we want to give a blood to the recipient. So the person AB blood group are called as universal recipient. They are received blood from person both any blood group, that means A, B and O. The person O blood group are called universal donor. They want to donate all the blood group to the any person. So it is called as a universal donor. So the antigen A, antigen B, antibody A and antibody B, all the seen in this. So next we go to see negative and positive, that is called as a RH factor. So this is discovered by Landsteiner and Weinzer in 1940 in the rhesus monkey. The surface of RBC contain antigen RH factor, that person called as a RH positive. So the surface of antigen, the RBC is not having a RH antigen, they are called as a RH negative. So person do not have a RH antigen on the surface of RBC. Antibodies develop against RH antigen, it's called as RH antibodies. So we cannot give how the negative to positive also. So we are finished the chapter, this lesson unit 2, that is lesson 14, the circulation in animal.